I got a bit of a cold for everybody who's here, so I might be clearing my throat a little bit, but otherwise I'll just be boogieing on through. Yes, so dancing in my dendrites, it's about doing everything you can that's possible and keeping your mind alive. There you go. Everything from balancing on curbs to very diverse things. Very, very important. We're going to be talking about, yes, the signature of this TBI. It's sort of a metabolic type of TBI. It's linked to Alzheimer's and also Parkinson's. We're getting more and more. You're going to see some cool stuff on Parkinson's coming up. And audiovisual entrainment. So a little quick history. I got involved in 1981 with the University of Alberta in dentistry, designing this research lab for them. And at that time, I started designing electrostimulators. I did some biofeedback devices as well when I realized that TMJ was a lot more than just a dental issue, but it was also a mental issue. So I started designing of my first machine. That's the big one here. The big one here was the first one I designed. I only made two, one for an instructor in performing arts who's using it to help his students overcome stage fright, and which is why I designed it in the first place. And I kept one for myself. And I didn't expect to sell more than two, honestly. It took so long to design because the guy didn't really know what he wanted. And I made $1 an hour over the course of 8 months working in my basement. We've sold 60,000 devices since then. And so I guess there's something to it. But I wasn't sure if it was real science or not real science at the time. I'm very convinced it's real science now. So as you guys know, if you're just doing a spectral analysis or spectral array of an EEG, you're going to see your big alpha frequency right around 10. So yeah, lots of course, typical alpha peak. And then we have a bit of a beta peak around 20-ish. And this is Thatcher's work. But then as soon as you start getting a mild closed head injury, the alpha comes way down, and so does the beta. And then when you start getting into, of course, the severe, you lose your alpha pretty much entirely, and you lose the beta, but you lose your alpha rhythm, and that's an important thing we're going to look at. And you get all this delta activity, though typically it's low voltage. I think that this graph is a little stylized, and really this line here should be half that, really, really low. That's what I see anyway. And the delta seems to only come because neurons, and especially in the nodes, they're just firing randomly, and they just show up as noise. And we'll get into that a little bit too here. So when pyramidal neurons are not modulated, such as with a thalamic sync pulse, like with alpha waves, they fire randomly in the delta band. And that messes up our EEG. We have to know how to interpret that, or we're not going to know what we're looking at. And this is one of the things I had to sort out when I had all this low voltage choppy stuff that was quite high in this low voltage chop in the beta bands. It was really delta activity from the various nodes getting vector added into the EEG and giving me fake beta is what it looks like to me. Anyway, some real quick stuff here. Only when it comes to MRIs, MRIs are not very good at looking at closed head injuries. I've had several clients who passed an MRI entirely and were told they didn't have a brain injury, but they certainly had a problem. EEG has certainly a better approach, but I think we need to learn a little more about how to interpret our here as someone with multiple TBI. They got a little bit of delta activity going on once in a while. They're very low voltage and choppy, and they don't have any alpha. And this is the whole head. Usually you see it in bands. Depends where the injuries are, but sometimes, not often enough, I see the entire head goes down and there's no alpha waves anywhere. So this person here, 54-year-old female, and she's all in the weeds in a sense. Now all these, of course, all their networks are affected hugely. And networks are interesting because if you thought of these as phone systems, so there's a phone system here with 1,000 lines on it or 1,000,000 lines on it, and there's 1,000,000 phones over here that you can call. They can talk to each other. If this side goes down, these sides have nobody to call. So they go down too. And this is some of the issues that we see. And it's called diaskesis. And this is it here. So diaskesis meaning at rest, meaning to cut at a distance. So here's a brain here. If they damage this part goes down too on the right-hand side. Functional diaskesis, when these parts are damaged, some parts get shut down and some parts get overexpressed, some parts get underexpressed. Same thing with the connectomal diaskesis and the connectional diaskesis as we see down below here. So things can get overexpressed because a lot of neurons are inhibitory and some are excitatory. And so you can have a damage in one little spot of the brain, and other parts of the brain go wild, and other parts of the brain get quiet. So here's a diffuse axonal injury here, and they're looking for these little tiny spots that show up where axons have been torn from someone who had their head jostled in an accident. And we're not looking at that. What we're seeing a post-inflammatory condition, and this is how I'm describing it here. So we've got normally the thalamocortical loop. So the cortex says to the thalamus, hey, how you doing? The thalamus says, oh, good, how about you? So of course the alpha waves, the thalamocortical loop, and same thing with delta waves, they're hyperpolarized thalamocortical loop when we're asleep and they're in the delta band. So default mode network is active. It sends a little signal down to the thalamus. Hey, how you doing? Yeah, I'm doing good. How about you? Yeah, I'm doing fine. How about you? Yeah, I'm doing fine. How about you? And it goes through these loops, at 10 hertz. So you get at least 2 action potentials every 100 milliseconds, if not more. Parts of the brain also go through the putamen and some go through the caudate. Especially the caudate will be involved in this type of signature, and I'll show you why later. But if you had, let's say we had 50 billion neurons shooting signals down to the thalamus and back, because the thalamus is innervated through most of the cortex, and you lost, let's say, two links each way for each cycle, and there are 10 of them, so that's 20 action potentials times 50 billion. I mean, you are losing over 1 trillion action potentials a second. Normally, when our eyes are open and we're processing, we've got cortical oscillations going on, but in this case, we're just losing the thalamus, and we don't have anything cortical going on either. And yeah, they're losing a trillion action potentials a second potentially. Now, here's something about alpha. Now, this is from the database, and if you look at alpha and beta at FP1, FP2, and as you know, of course, as you get towards the back, the alpha beta ratio alpha beta ratio gets stronger and stronger. So it's not too high at the front on the forehead, but as you get towards the back, it gets a lot higher at 4.4. And this is going to be important when we're interpreting the EEG. So I use the skill, for, this is Sturm and Kaiser's image database. I think it is one of the better ones that were ever made. And unfortunately, with Sturman's illness and in passing, of course, and David Kaiser, just kind of being the guy he is, they never really promoted their database like they should have. And so other guys are promoting their databases a lot more vigorously. And the skill has kind of disappeared from existence in our profession. But phase is absolutely critical in this condition. You get phase issues when you have random delta firing in those sections and then in the nodes, and you get phase issues as you start to demyelinate from a lack of action potentials up in the beta bands. And most databases don't look at phase at all. I'm disappointed that WinEEG doesn't look at phase, and I use WinEEG, but it doesn't look at phase. It does some other things that are pretty cool, but it doesn't look at phase. The NeuroGuide, yeah, it kind of does, but at least it does to some degree. But I still think the scale probably shows the best phase, and we'll see some of that later. So anyway, TBI, effects on brainwaves. This type of TBI is not caused by concussions. Most people get a concussion, they're off work for a couple of weeks, then they go back to work and they're doing reasonably well. But then a couple months later, they're not doing so well. And then 3-4 months later, they're doing really poorly. They start developing severe insomnia. They're exhausted all the time. They start to lose their cognitive abilities. They start to lose emotional stability, and they start bursting out in anger and stuff like this. And then they're unemployable 3-4 months later. And then they start, and then they have chronic anxiety, chronic insomnia, and they all start becoming obsessive compulsive. So my group that I see, they're all hoarders, counters, cutters, ritualists, door closers, tappers, anorexics, and committed substance abusers. And they all show an OCD pattern, but not a typical OCD pattern. So anyway, what we think is going on is there's an interruption or a disconnect that's going on, probably caused by reactive gliosis. And the brain maybe is taking neurons offline as a way of a protective mechanism, and then forgets they are there. Because I tell you, the brain needs chemical gradients and electrical gradients and stuff for everything to work together. And when the glia starts to lose its calcium gradient and other gradients, it starts to go down. And when it goes down, the neurons go down because they're missing a gradient from the glia. And there's this cycle that spirals downward. So the frequencies of alpha are set in the thalamus, as we all know, and the thalamus gates perceptions into perceptual packets. And I'm not going to really get into this too much, but we start to lose all of this kind of thing. So the thalamus can provide rhythms in the range of 3 to 16 hertz, with the common range of being 8 to 12, which is alpha. And hyperpolarization of the thalamus slows the alpha, and hypo speeds it up until it desynchronizes at about 16 hertz and becomes a low voltage fast EEG. That plays out. But glutamate can trigger actively propagating calcium waves in the cytoplasm of astrocytes. The astrocytes, the microglia, the oligodendrocytes, they're all using signaling in different ways from the neurons. And the neurons also need signaling back and such. And they also need to keep the mitochondria going. And if the glia does not know the neurons are there, it doesn't help nurture the mitochondria and keep the ATP rolling and stuff like this, so the neurons can function. So astrocytic calcium waves are present under these circumstances, and these waves can be triggered by the firing of glutamatergic neuronal afferents with latencies as short as 2 seconds. So stuff can happen quite quickly in the brain, even on a chemical, even from a chemical perspective, things can happen quickly. The ability of tissue astrocyte networks to respond to neuronal network activity suggests that astrocytes may have a much more dynamic and active role in brain function than has been generally recognized. Okay, calcium is a universal second messenger that regulates the most important activities of all cells. It is of critical importance to neurons as it participates in the transmission of the depolarizing signal and contributes to synaptic activity. Neurons have thus developed extensive and intricate calcium signaling pathways to couple the calcium signal to their biochemical machinery. Abnormal calcium signaling and its involvement in synaptic signaling processes, neuronal energy metabolism, and neurotransmission in the most important neurological disorders must be considered. There you go, that was a mouthful. And I'll get through some of this dry physiology fairly soon. So if the connectome represents a complete map of anatomical and functional connectivity in the brain, it should also include glia. Glia define and regulate both the brain's anatomical and functional connectivity over a broad range of scales, spanning the whole brain to subcellular domains of synaptic interactions. Glia interacts with the neuronal connectome, including long-range networks, which I think we're generally not really taught that so much in our neurophysiology, but local circuits, individual synaptic connections as well. And our understanding of the structure and function of the neuronal connectome would be incomplete without an understanding of all types of glia contribute to neuronal connectivity and function, from single synapses to actually long-range circuits. So astrocytes, fast local electric field oscillations in the gamma frequency band have been closely correlated with learning, memory storage and retrieval, attention, and other cognitive functions. This frequency band comes about from healthy mitochondria in glia. And now this is really important. I've been doing a lot of research in mitochondria, and so many things take down mitochondria. The list is endless. Excess sugar, artificial sweeteners, artificial colors, artificial flavors, pesticides, herbicides, all these things take down mitochondria. But the latest research I came across just a few months ago was pretty scary. This group they have developed a microscope that can see from micrometer to nanometer particles. And they were looking at bottled water, and they were finding up to 100,000 nanoplastics per liter of water. And a liter is about a quart, in the American measure. That's pretty close. And 100,000 particles, they all cross the blood-brain barrier, and they all cause dysfunctional mitochondria respiration. So even something as simple as bottled water could eventually cause our brain to go down, and shows the importance of keeping things pure. So anyway, so astrocytes, they're placed to modulate neuronal activity, and mitochondria are absolutely critical, and everything we touch or eat can affect it. So here's an example here. You can see here the neurons are in yellow and the astrocytes are in orange, and they are proactive with those neurons, keeping them fed, keeping them nourished. all the time. So anyway, as we mentioned before, when parietal neurons are not modulated with a thalamic sync pulse, they fire randomly in the delta band. Now, when we look at the rich club nodes or the Hagman hubs, I think that we're seeing some issues with this when we do an EEG, because I've been getting these low voltage choppy, as we saw earlier. And we'll look at more of this again. And I think what's going on is when they lose the thalamic sync pulse and they start to fire randomly in delta, here's what's going on. Normally when we have a sync pulse, like say with an alpha wave or even with a delta wave when someone is sleeping, but the pacemaker is the thalamus, or the loop between the thalamus and the cortex, and it's firing off. Billions of neurons are firing all at once. So we see a large, nice, beautiful wave on our EEG. If supposing that sync pulse wasn't there and the neurons are now firing randomly. So imagine this, I'm going to clap my hands a bit here. So imagine we had 100 billion neurons all firing, or 86 billion neurons, whatever, all firing together and at 2 hertz. And you would see a giant 2 hertz signal. And imagine if you had a microphone in a room and there's a crowd of people all clapping their hands to a song that the band is playing, and they're all clapping to the beat of the band. Well, you'd have this big spike showing up, let's say at 2 hertz, let's say if you're clapping kind of like this. But at the end of the song, when everyone applauds, all that 2 or 3 hertz clapping becomes desynchronized because they're not clapping to the beat of the music anymore. They're just applauding the band. And once it becomes desynchronized, it turns into high frequency noise. And I think the EEG is picking up this desynchronized delta and it's seeing it as high frequency noise on the EEG. And here's another example of the rich clubs all firing together. Well, that's what I was just explaining about. So move on. And of course, you're going to get extreme phase issues all over the place. It's going to look more like noise than it is going to look like synchronized beta anyway. So neurons and oligodendrocyte precursor cells establish functional and structural axon glia synapses with each other in both gray and white matter. Now, oligodendrocytes don't use chemical signaling, as do other aspects of glia like astrocytes. They're looking for electrical gradients. And if they don't see electrical gradients, they quit myelinating. Some interesting research on, and here's an example right here. So it's picking up these signals from a neuron and it myelinating the sheath. So if we lose 1 trillion action potentials a second, what happens? Now, this is an interesting study here. So when they place animals into an enriched environment, it automatically increases proliferation of the oligodendrocyte precursor cells and the development into myelinating oligodendrocytes throughout the amygdala, hippocampus, cortex, and white matter. So everywhere. Every time we learn something new, we end up thickening the myelin sheath on those neurons that are involved in the new skill. Every time we lay around on the couch for a week and do nothing, the brain starts to not myelinate and the myelination starts to thin. So that's partly why my theme is dancing in my dendrites. It's about keeping as much of the brain myelinated as we can. So I do all kinds of crazy things to try to keep my entire brain myelinated. And for these very reasons, although I would do it anyway. But so in gray matter, astrocytes export lactate from glucose or glycogen to neurons to power their mitochondria. And of course, if astrocytes aren't working right, because someone's drinking a lot of bottled water and eating seared meats and artificial sweeteners and colors and all that stuff, and their mitochondria going down in their glia, well, then it's also going to be taking it down in the neurons because the glia nurture the neurons. So in white matter, lactate can support axonal functions in conditions of energy deprivation. Oligodendrocytes are the cell type in the brain that consumes lactate at the highest rate. So very, very important. Lactate can support oligodendrocyte development and myelination. Everybody still with me? I know this is a bit dry, but we'll get through this. Activity-dependent voltage-gated release of calcium at axon and oligodendrocyte precursor cell synapses affects, I'll just call it OPC and myelinating oligodendrocyte development. Proliferation and differentiation of OPCs and myelination of axons by MOs can be modified by neuronal activity in vitro and in vivo. So they actually did electrostimulation of different frequencies and they were seeing how that affected oligodendrocytes, and so on. And you can see here, stimulating at 25 hertz and 300 hertz resulted in higher mean density than 5 hertz for dendritic, myelination. Okay, so yes, so as I was mentioning before, about 1 trillion action potentials might be getting lost when we close our eyes, and we should be making alpha waves, and we aren't. This is also something I've been wondering about. Is MS maybe a part of this? And this whole thing going on, and so I'd like to do more research in the whole MS side. Now, corpus callosum is interesting. You're going to see this in some of the EEGs. So myelinated nerves can send a signal across from left side to right side or right side to left side within 30 milliseconds. And 30 milliseconds roughly equates with about 30 hertz. 10 hertz is 100 milliseconds. 30 hertz is 33 milliseconds. So right in that beta band, almost low gamma band, non-myelinated nerves take all of a third of a second to cross the corpus callosum, and that works out to about 3 hertz. So as we lose our myelination, we are going to start to see slowing in the beta band, and we're going to also see some severe phase issues between the affected circuits and the unaffected circuits. So I don't know if reactive gliosis and inflammation, if the neurons that were out of sync lost their link, or the brain drifted electronegative, as with thalamic gating, and squelched out the thalami. So I bought a DC amplifier to find out. Now, taking DC recordings, it turns out to be extremely difficult. A proper DC recording is very, very difficult. And you need to have impedances in the 500 ohm range. And you need to draw blood a little bit so that you can get rid of capacitance under the electrode. And so I don't get too many people who are willing to let me dig electrodes into their skull so hard that they're bleeding a little bit under every site. So this is a lady here, and she was willing to cooperate, partly because her job was on the line. When she goes to work, every once in a while, she gets severe headaches. Well, pretty much every day, and nausea and so on. And she believes that the lights in her office are doing it. So she came into the office, and we had fluorescent lights in my brain room, and turned the lights on, see what her brain did. Turn them off, see what happened, turn them on. And her eyes are open, by the way. So she's not going to be making any alpha waves per se. And about every 1 in 3 times that we turned on the lights, her brain went into this weird oscillation, big spike and wave stuff. Kind of a seizure almost, but not quite. But certainly paroxysms, and they would linger for some period of time, and then she'd feel very out of sorts when this was happening. So we're looking at this, and we're doing a DC recording. On the EEG, we can zoom out and we can actually see the DC levels. Now, DC is representative of not of electrical activity in the brain per se. It's representative of the chemical activity in the brain. Your glutamate, your ATP, your calcium gradients, these all have charges on them. And what we can see here is that she's well into the millivolt range, not the microvolt range, but the millivolt range. She's like 2 millivolts. I had it set to -1 just so that it would really be nice and black, so it was easy to see. But truthfully, she was about -2 millivolts, so almost 1,000 times higher, certainly 500 times or several hundred times higher than what you'd expect from EEG. And look how it correlated with her slowed activity, which is more in the occipital parietal area. And even up here where we're a little bit spun off into kind of F4, F8 area, we see that there is negativity going on there too, affecting that area. So there's definitely something going on a DC and a chemical level that is influencing the sort of the hyperpolarization of the thalamus to the cortex and slowing everything down. But these are really difficult recordings to get. We need a way to jumpstart the brain. So thalamically focused ultrasound now you're going to see more stuff on ultrasound. Ultrasound is becoming a thing. But this was done long ago, and they found that when they fired ultrasound at the thalamus, they could reduce anesthesia time in rats by activating the thalamus. And then they did some stuff after severe brain injury they worked it on some people who were in sort of in a semi-coma and got them back awake. So that was interesting. So what happens if we take light and we chop it up into little bits? So instead of just being continuous, it's starting to flicker. And that's kind of what the slide is depicting. And of course, this is what our gear does. Our gear is meant to chop up light, continuous light, and make it into all these little bits and shoot it at the eyes and see what the nerve input to the brain does. So here we are shooting it at the eyes. And we can do 2 frequencies with our gear, which I think is important. We used to think the 2 frequencies was really important from a frequency model, but truthfully, there's so much more going on that the frequency model of entrainment is rather, it's not that important. When you consider that all the things that entrainment does, you can do frequency driving to some degree, but you also affect cerebral blood flow. You also affect lactate and ATP. We also affect heat shock protein 70. We also affect inflammatory cytokines. We also affect cerebrospinal fluid circulation. A lot of things go on with entrainment, so much so that the frequency part of entrainment is really not that big a deal. All right, here's an example of some of the things that we do affect with entrainment. I mean, when they discovered entrainment in 1934, they only saw the frequency effect on an EEG. They didn't have the ability to test these other things. But it does so much stuff that is not frequency based. In fact, we have found that dirty stimulation or mildly randomized stimulation is somewhat more effective than a fixed frequency. And so many people doing neurofeedback, because neurofeedback is a frequency model, they want to know a specific frequency for this and a specific frequency for that. I'm saying there isn't one. In fact, muddier works better, generally speaking. Just don't be too random or it doesn't work at all. But mild randomization works much better than a fixed frequency. So because we're affecting these other systems. Now here's some quick stuff. So this is an example of frequency model with a fixed frequency. And you can see at 8 hertz, we are really driving up 8 hertz throughout the brain. Frontal, central, parietal, occipital somewhat. Not so much in the temporal lobes, but we are really driving it up. Here's another example of someone who is really deficient in the SMR band, high alpha SMR band, 12 to 15 hertz roughly. We gave them stimulation kind of in that band, and 20 minutes later, it's all cleared up. This is a study here, which I wasn't going to show you, but what the heck, these guys were using entrainment under the premise that theta should improve memory, although alpha improves memory, SMR improves memory, beta improves memory, gamma improves memory. So it's hard to say, but the theta, there's that theta premise that if you're firing in the theta band, you'll improve memory. So these guys are using about 5.5 hertz, and you can see the theta power is getting distributed frontally, and their memory also improved a fair chunk. And I'm not going to get into this. That's in another lecture, really. Here's some stuff on cerebral blood flow, and when you get a person relaxed and calm down the HPA axis, hypothalamic pituitary adrenal axis, you can get hand warming. And of course, all the autonomic stuff kicks in, right? Same, here's cerebral blood flow. Now, cerebral blood flow, this was Fox and Rakeley study. They found that roughly 8 hertz was maximal cerebral blood flow produced in the brain. And they found that 5% increase, there was the whole brain had increased cerebral blood flow by 5%. And then other studies on cerebral blood flow have come out since then. This one here looked at 2 hertz and at 8 hertz, and they're looking at the changes in cerebral blood flow, and also, which is in black, and also cerebral blood volume, which is the gray, and in response to visual stimulation at 2 hertz and 8 hertz. Cerebral blood flow, very important. And it plays out in other things that go on. Now, this is a study done by Sappi Mariner long ago on looking at lactate and ATP production in the brain using visual stimulation, flicker stimulation. And we can see here, when they turned on the lights, lactate and ATP went way up. And when they turn the lights off, lactate and ATP went down. They turn on lactate and ATP go back up again. And these are important for all those disorders where lactate and ATP have gone down, probably due to mitochondrial issues. This could also be tied into maybe why red and near-infrared photobiomodulation might be affecting lactate and ATP by stimulating. There's lots of ways. Methylene blue is a nootropic that definitely stimulates mitochondria. And there's plenty of people out there who take low-dose methylene blue to really sharpen their minds. I take methylene blue myself, and it's pretty amazing stuff. What it can do to your brain. So astrocytes produce and release L-lactate as a source of energy for neurons. L-lactate serves as an astrocytic signaling molecule in the locus coeruleus. That's norepinephrine. The locus coeruleus is the main source of norepinephrine. There you go. And it modulates the brain, and it certainly modulates our alpha waves. People who are overexpressive norepinephrine can have fast-running alphas. In fact, I have seen this with people with subluxated atlas bones. When they've had a fall, don't have to have a concussion per se, but a fall where their head gets jostled in just the right way that the atlas gets pushed to the side, pinches the brainstem, and causes all kinds of neurological problems. The worst case I saw was a boy who was paralyzed roughly half an hour every single day after just a mild accident that neurologically had no effect, but he popped his atlas bone out. And he flew up here from Sacramento. I was lecturing down there, and his father took my lecture in, and they came up, and I looked at him, and I knew him within the first minute that he had a subluxated atlas bone just by his posture and how crooked his shoulder and neck was, and everything else associated with that. You can just tell by looking at a person pretty much. And we sent him straight off for NUCCA, NUCCA, National Upper Cervical Chiropractic Association. And so far, I have found that they are the only people who can reset an atlas bone. Well, anyway, they reset his atlas bone the next day, and he never had a paralysis. He was never paralyzed again, and he went back to school, and he got his license back and had a completely normal life just from resetting that. Meanwhile, they had spent $120,000, $20,000 in copay, and the insurance company picked up the other 100 with standard of care, who did nothing for him at all. In fact, they were going to fuse his bones together in his neck and would have destroyed his life. He just needed a little adjustment, and the NUCCA guys are the only guys I find who can do it. And all my concussed people nowadays, they must have a NUCCA assessment before I will even do an EEG. And I got a whole lecture on that, if you guys want to see that down the road. So lactate in the brain, it's relevance to brain energy and so on. So lactate from astrocytes may be crucial for supporting axonal function, especially during times of high metabolic demands or hypoglycemia. The astrocyte neuron lactate transfer shuttle system. Now, there's a bunch of different transfer shuttle systems. There's an electron transfer shuttle system in all mitochondria as well. So it's not just with lactate, but it's also with electrons. And if the electron shuttle system is down in mitochondria, the lactate transfer shuttle system will also be down in the astrocytes, which in turn will take it down in the neurons. And so you end up with a cascading effect, a snowball effect. So anyway, it serves as a protective function to ensure a supply of substrates for brain metabolism, and oligodendrocytes also appear to influence the availability of lactate. Of course, they demand lactate, as we saw in the earlier slide. There is increased evidence for lactate acting as a signaling molecule in the brain to link metabolism, substrate availability, blood flow, neuronal activity, and panic disorder. And we see enough people with panic issues. Okay, heat shock protein. Heat shock protein is known to protect the brain from injury through multiple mechanisms. This is a study actually done on one of our little devices, might have been our David Paradise device, but they pushed up heat shock protein 70 by 1.84 times. Well, I should have mentioned up here too when we looked at this one. Basically, flicker stimulation pushed up lactate and ATP by 260% in about 5 to 6, 7 minutes. Very quick. So that's something good to know. And heat shock protein went up by 180%. So with entrainment. So yeah, and they were just using, I think, 8 hertz in that study. So it's pretty wild what things can do. So anyway, heat shock proteins, they're all over the brain. They help protect it from viruses, cancer cells, they boost immunity, and so on. And heat shock proteins can be triggered, by the way. When heat shock proteins were discovered, they discovered it by making things, the living organisms, very, very hot. And they found that they liberated heat shock protein to protect the cells. But they also found that extreme cold will also trigger heat shock proteins. They should maybe call them just shock proteins. And other things, stressors can generate heat shock proteins in general as the body tries to protect itself. So here it was, 8 hertz. Yeah, okay, by 1.84 times. with entrainment. Now, entrainment also affects the P300 ERP. And this was go, no-go test. This is John LeMay's work in 2021. And he found that when it was a go test, and because everybody's getting into ERPs nowadays, and it was a go test, the strength of the ERP was much higher after entrainment than it was with the controls, because the neurons were firing much more in unison after entrainment than they did prior to entrainment. When you looked at the no-go test, which demands more energy and effort from the brain, the controls did not do so well on the post-test, and the entrained group did much better in terms of that amplitude of the P300 response and a very consistent latency across the entire brain. This study was done by a group out of MIT, and they had a whole bunch of mice that were, I'm not a real fan of this study because I don't know if it really reflects true Alzheimer's. But what they did, though, is they had these mice that were genetically transgenic mice designed to make plaques. They put them in a box, one group anyway, in a box, and they flashed the lights at 40 hertz. And later that day, when they sectioned up the mice and compared them against the control mice, they found basically, well, I'll call it 60% reduced amyloid beta deposits in the brain. They also looked at the microglia cell diameter, and it increased by 166%. That's this one here. And they also looked at the amyloid that was in the microglia itself, the amyloid deposits in the glia. And the glia got fat because it was gobbling up amyloid, beta amyloid deposits. And 60% reduction in one day. So that was pretty impressive. And so they're using that now. They got a patent on it, and they're going to start trying to promote gamma stimulation, I think, in their own device, so that we've been doing stuff like this for 30 years ourselves. But anyway, away she goes. Now, cerebrospinal fluid flow, fourth ventricle, really is being jumping out as in research these days with Alzheimer's and also in Parkinson's. And what they're looking at here is this here is the guy on the right is healthy, the red, and the guy on the left is not healthy. Red is more fluid flow than the yellow is. And you can see it's a much larger spot here and right here at the ventricle compared to a normal person, Alzheimer's person here, and much tinier. So, and here's the thing, there's the lymphatic system, and what they're finding is that delta waves cause the microglia and the lymphatic system and they can pump a lot of deposits from the brain out. But if a person has impaired sleep and does not make those proper large delta waves, the pumping doesn't occur, or it occurs very poorly, and the brain fills up with deposits. So sleep is important, and every single person I've got with this brain condition cannot sleep. And that's why it turns into Alzheimer's or chronic traumatic encephalopathy over time. Now here's cerebrospinal fluid flow in ventricles of Parkinson's. Similar idea. They looked at more nodes, more ventricles, not just the one in the back of the head, the fourth, but the third and the second, the lateral ventricles to some degree. And here's Parkinson's here, and here's the controls. And the controls are much redder than the Parkinson's, and that indicates increased cerebral spinal fluid flow. Now I'm going to show you some really cool stuff. So fMRI was used to simultaneously measure blood oxygen level dependent, or BOLD, signals, as well as cerebrospinal fluid inflow in the fourth ventricle. And visual stimulation was in the form of a checkerboard pattern. They had different frequencies where they were altering the pattern: 4 hertz, 8 hertz, 12, and 40. And then they did a couple of different studies. So they did 2 experiments. They measured neuronal, hemodynamic, and cerebrospinal fluid signals during visual stim using simultaneously EEG and functional MRI at high temporal resolution. Experiments 2 and 3, they were using independent fMRI data sets, and they were used to replicate findings from experiment 1 and manipulate stimulus parameters to test whether this pattern was consistent, blah, 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 with neurovascular coupling. And maybe I'll skip some of that because we don't need to take all that in. But here's the pattern on and off. Now they tried different rates. Like this is 16 seconds on, 16 seconds off. They also tried 8. They tried, I think, 30. And they found that 16 seconds on and 16 seconds off was the best for visual stimulation. And Here's what they found. Well, here's where they were taking their slice, and there's the fourth ventricle down here. And so they're seeing the cerebrospinal fluid going in and out. Now, the way this works, and this is really clever, is that when you have intense stimulation, cerebral blood flow goes up dramatically. But the brain has to maintain intracranial pressure, so it has to dump cerebrospinal fluid. So it pushes it out through the fourth ventricle. But then when you turn the stimulation off and the cerebral blood flow drops, now it needs to pull cerebrospinal fluid back in. So it turns out the more stimulation you get, the better circulation you get of your cerebrospinal fluid. And delta waves also cause this, but just having an active life and stimulating your dendrites in all kinds of ways are important for maintaining circulation of cerebrospinal fluid. Use it or lose it kind of an effect. So in experiment 2 here, what we're looking at here is when they turn the stimulation on, cerebrospinal fluid is flowing out. When they turn it off, when they turn it off, it's flowing in, and when they turn it on, it's flowing out. And it's quite significant. And they found the entire brain did this, not just the visual cortex, but the entire brain did it. Here's another example here of the different frequencies. These are the stimulation durations. And they ended up doing 0.2, 0.5, 1, 2, and 4 seconds on, 4 seconds off. And then they looked at this again up to 40. And they found that 8, 12, and 40 hertz of flicker stimulation actually produced the best overall cerebrospinal fluid flow. And so that's what they were doing here. So it's pretty easy to make our machines do 16 seconds on and 16 seconds off at 8 hertz or 12 or 40. That's easy to do. And we have a new device we're coming out with fairly soon. And we're going to be making some pumping, cerebrospinal fluid pumping protocols. We have a belief that it pumps anyway, especially when you have dirty stimulation and you've got latencies between left and right visual fields going on. There's probably a pumping action that occurs anyhow. And sometimes the sub-delta one works extremely well for people, and maybe it's doing a pumping action. It never occurred to me before until I read the study. But we're going to make some pumping ones based on 8, 12, and 40, and I'll get them in our new device. It's called the Premier, and it'll be coming out fairly soon. So randomization, I mentioned that when we randomize slightly, we actually get better effects than a fixed frequency. And it can be a little frustrating. All the neurofeedback guys always want to know a fixed frequency for doing neurofeedback, when actually we're looking at all these other systems that are not frequency based, but they can influence frequency. But anyway, watching TV is audiovisual stimulation. A fixed frequency is audiovisual entrainment. But we found, now we did this work with Ed Pygott, Greg Alter, and another psychologist. This was done back in 2000, right around 2000, we started doing this research into randomization, and found that randomizing up to and anything in between 1 hertz, plus minus 1 hertz. So if you're doing alpha at 10, bouncing it around between 9 and 11, and bouncing the right side random from the left side as well. So if you're at 20, then you'd be bouncing around between 19 and 21. And that actually increased the effectiveness of the entrainment than using a fixed frequency. Randomizing at plus minus 2 hertz or up to plus minus 2 hertz doesn't work as well. And randomizing at plus minus 3 doesn't really do anything clinically. I gotta say, sometimes it does, but for the most part, it doesn't work well. So we've settled on plus minus 1 when we do our randomization. Some quick stuff, some cases we've collected over the last 20 years, looking at SMR data, randomized compared to, which is yellow, compared to fixed frequency, and for concentration, grade point average, memory, and worry, a randomization actually was more effective than a fixed frequency. So same thing with depression, balance. This was with seniors, the CANs, the memory, and peak alpha frequency, a little better randomized than it was fixed. Same thing with attention in seniors. This is microcog, attention, reasoning, spatial ability, reaction times, processing speed. And this was Tom Budzinski's stuff. He was one of the first guys to use randomization way back in like 2000. And we're like, whoa, that worked really good. That worked better than the fixed frequencies that I was using with seniors. So some interesting things come when you randomize lightly. Again, attention, impulsiveness, hyperactivity, variability, and ADHD. So all this randomization, mild randomization, where it's still mostly in training, You can still generally see it on EEG, although it's kind of muddy because it's not a fixed frequency, but it works. So because it's randomized AVE, we now call that RAVE, R-A-V-E, RAVE, randomized AVE. So that's what we've actually had a certain amount of RAVE included all our sessions for the last 10 years. On our new machine, I've programmed them to be RAVE across the entire session. And we've had units out for about a year now doing beta testing, and we're getting very good feedback from doing the RAVE approach across the entire session instead of just portions of the session. Anyway, here's some stuff here. Neuro 40 Hertz. This was, what's her name's work? Not Carter, Singer. And they were looking at these different pathways and how it was going to affect them and so on. And yeah, I'll skip that. Those are just some of the studies. We've done a lot of stuff with sports. Of course, sports people need good functioning mitochondria. Rocky Thompson never could win a game, and then he used our gear, and 2 weeks later he beat Lee Trevino, first time ever, and made a lot of money. Glenn Eller was 17th place in the Olympics, and then he got put on one of our devices about 2 to 3 months before he went to Beijing, and he got gold. It just stabilized him that much. We've been working with this young lady, Marissa Ponich, for a long time for fencing in the Olympics. This lady here used entrainment. A lot of high performance athletes, when their career is done, develop severe depression. Their life suddenly changed. All this work they've done ends. They've been isolated from family and friends and relationships for 10 years, and everything comes to a crash. And this lady, that happened to her. She crashed. In fact, most of her swim team, this is the synchronized swim team, and they were in Australia. And after their career ended, she put on 100 pounds of weight and really struggled with depression. And then she used entrainment, was one of the, so I don't know who got her on the entrainment. It's in her book. I don't remember. But they got her on entrainment, and it started resolving her depression to the point where she could cognitively get involved again in her life and get her life in order. My daughter, the young lady in the middle here, she's been using entrainment ever since she was in grade 1. And when she graduated as a speech pathologist several years back, her marks were so high in some of her courses that they had to get Dean's permission to give her a grade, because otherwise it might look like collusion. And so, yep. So anyway, very proud of my daughter. Always gets me a little teary-eyed when I talk about her. So anxiety and OCD. Now this is important because this plays out with this signature a lot. So anyway, all these issues come out. Now let's take a look here. So here they're doing a slice of the brain right through here. And this is that slice. And we see the caudate nucleus, we see the putamen, we see the thalamus. And so cingulate goes through the caudate on its way to the thalamus. There's more connections in there than just straight to the thalamus, like you might see more in the parietal cortex. So there's more connections in there, and caudate is well implicated in OCD, obsessive compulsive disorder. I'm proposing there are spectrum disorders associated with this thalamocortical disconnect, and there's millions of people falling through the cracks. If you get a concussion and then you develop this, people kind of understand, and they're more forgiving because they know you were normal before the concussion. But people can get a virus and just have a bad flu, and it'll take them down just like a concussion. Okay, here's a hoarder. Now this is what I'm going to call normal OCD. What we're going to see with normal OCD is we're going to see that the alpha band right here down the middle, so FZ, CZ, often PZ, is higher in amplitude than the other channels. And this is what I would call normal OCD. And there it is down here. It's usually a little slow. And this I would treat with transcranial DC stimulation. Entrainment might work some too, but TDCS I think is more indicated for this type, where they make normal alpha waves, but they're in the wrong place. And here's this lady again on the database. And we can see that cortisol, she's had a traumatic childhood that went on for many years. And so cortisol has knocked down FP1, FP2, which is known as ventral medial depression in psychiatric circles. And she shows some of the asymmetry there as well. And we always see severe phase issues. Now, with everything we know about delta phase and neurons firing from the Rich Club on their own terms, so you looked at delta theta mostly. Didn't occur to me to look at beta, which is silly of me. But once I learned some things about beta, then that was the story. So here's a tapper. He has to tap his fingers sometimes for as long as 15 minutes before he can get himself away from it. He kind of believes it'll give him good luck, but he has to tap it just right, or he can't stop. And no depression or anything, but got this issue. Now, this was probably a thalamocortical disconnect from what I can see. Very choppy, low voltage. Again, these are probably the Hagman hubs or the Rich Club nodes, all firing delta randomly and showing up as choppy beta, is what I suspect. But every once in a while, maybe they do kind of fire together. And these guys can't sleep. And I did, of course, I didn't know this signature in those days. We were still learning it. Here's his OCD. Now, if I zoom out, you'll see OCD 6, 5, 4, 3 hertz. So they don't have any breaks in their cingulate and probably in that little caudate that's tied to it. If we take a look at them on what's called an entropy, which is more of a relative measure, we see the alpha band is gone. Now, some of this delta here is because of the fact that when the database was made, they were clipping it at around 3 hertz. And they had a filter that was, I think, 3 dB at 3 hertz, and then it rolling off at 6 dB per octave from there on in. And the amplifier I'm using, of course, goes to DC, and I normally set it to half hertz, but I want to see more. And so it's going to overexpress itself in the delta band. But I don't worry about that as long as we know why it's happening. That's what really matters. The important thing is here is that we see they don't make any alpha. Now, we do see up here some stuff going on. That's probably a little bit of EMG. In fact, it's hard to say. Yeah, might be a little EMG back there. So anyway, here's the phase issues. And we often see corpus left-right issues going on. This would be probably corpus callosum or other networks that cross from left to right. And this is now the first time when I really connected the dots with entrainment. Now, here's the clincher. I showed you those ultrasound studies and how they were using stimulation to reactivate a thalamus. Well, they were using beta frequencies, not alpha frequencies, but beta frequencies to activate the thalamus. For the longest time, I was doing the neurofeedback model, and I was using alpha to try to reactivate their alpha. And it didn't work. I mean, it worked on occasion, but not very often. Then I started experimenting with blended frequencies. If they had some depression, I did alpha beta. If they had otherwise, I just did an SMR beta. And I had suddenly much better results. And then when I randomized it, some I had even better results again. So here's a lady here with she's anorexic. And you can see she had a flu that was really severe. And then after that, 6 months later, she started becoming anxious and obsessive. It takes a while for this process to occur. It's usually not right away. Typically, we see 6-month delay before it happens. Struggles with anorexia, and you're going to see this with COVID. Struggles with anorexia, has high interpersonal anxiety, very poor sleep, and they all have very poor sleep. That is definitely something that stands out in everybody. And we can see here, pretty choppy, low voltage, occasionally makes a bit of alpha here and there, but for the most part, there isn't much going on. Here's the OCD, boom, 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 all the way across those bands. They don't have any breaks, and so the cingulate and the caudate go wild. This was looking at her magnitude of her alpha straight on. This was before I was using entropy and relative measures. Relative measure, you'd see these much bluer in the 9 to 11 hertz range. We see that there are some issues going on in delta theta with regards to her phase. This study kind of says what's going on. So disrupted gamma synchrony after mild traumatic brain injury and its correlation with white matter abnormality. Now, fortunately, I kind of figured this out right around this time on my own. Well, maybe it was from the study. I can't say with certainty, but definitely something that woke up my eyes to, I should be looking at beta phase. And this study certainly is part of that. So take a look at beta phase with this girl now. And whoa, am I seeing some issues? Yeah, things are really messy here. And yes, so definitely, I don't know if we're looking at necessarily myelination issues, or we're looking at general transmission velocities that are related to other things. But nonetheless, as we see in this study, it is definitely there. And it's an issue that needs to be resolved for sure. And these guys here, which we talked about earlier, you can see what's going on. So I put her on the entrainment. I was using a randomized alpha beta, and started getting these alpha spindles, massive alpha spindles. This is 20 minutes into the session, massive alpha spindles. And she fell into a deep sleep and slept for half an hour after the session ended. And here she is, fast asleep in the chair. That's my old system before I went with a fancy new DC system, a wireless system from Mitzar called the, well, I forget what it's called. It'll come back to me. But anyway, I got a nice little tiny system. It only measures about 2 inches by, or maybe 3 inches by 2, and it's about a ½ inch thick. It just plugs into the pigtail and transmits to the computer, and it's a very nice system. So here she is. Now you can see here she's at 5 microvolts now. Before she was at about 2 or 3 microvolts with her on her raw EEG. Let's go back up to, I think here, 1.8 microvolts. So now we're at 5. And the biggest difference that you're going to see at the moment, yeah, she's still showing some of that OCD pattern, but she is also now making alpha waves. That's all cleared up. And what I'm finding is the ratio of the OCD versus the alpha waves is a really big indicator of OCD. That's probably one of the bigger indicators of whether or not a person's going to have OCD, is that ratio. Of course, if this pattern is high along the cingulate, so if you have low alpha and you have severe anxiety and you're obsessive compulsive, it's just going to amplify that so much more. As soon as we bring up alpha, the OCD tendencies go right down. When we look at her on the database, now this happens a lot. Initially, when we fire people up on the database, their alpha is running at 13, 14 hertz. It's like it's being influenced by a lot of norepinephrine. I don't know why it does it, but over the weeks, it slows back down to 10, 11, and it'll sit there. Here's her phase issues, and they've improved a fair chunk, and so on. So she put on weight. She's an 18-year-old female, I should have mentioned that. Anxiety was gone. She slept 10 hours that first night, and she has been sleeping 8 hours ever since. They have to take the unit home and use it every morning is what I prefer, right when they wake up, so it gets their day in order. And normally after about 3 months, the brain gets established, and they don't need to use it anymore, but they often still will use it for maintenance. If you're having a bad day for some reason or a lot of stress, I mean, all of us have bad days once in a while, and entrainment really helps if you have a bad day and you're stressed out for whatever reason or something bad happened in your life and you need to kind of keep stable. This is her SCL90 profile. You can see somatization, obsessive compulsiveness, all very, very high. Anxiety is high. 50 is the norm for the SCL90R, and 10 points is a standard deviation. She's 70, so 77, so almost 3 standard deviations off. 3 weeks of using we put her on beta. We started her in my office. She was on alpha beta, but we put her to SMR beta, and her score was now 5. And we see that a lot with this population. Here's another lady. She's 5 years. This lady here, 5 years. You can see she's definitely low voltage, choppy. And we have people who can't function at all. And when after entrainment, they go back and get their college degrees. They improve so well. So here we go. She had a closed head injury. She couldn't distinguish between right and left. So she definitely had visual perception issues. She had balance issues. Strange things were happening with her vision. There was delay in seeing things. She couldn't work, and she was bumping into things. Again, poor sleep is always and also the somatized. So they have, in this case, very high pain sensitivity from her car accident. And there she is. A lot of eye tics. It's usually a sign of anxiety as well. Take a look at her here this is 2 standard deviations on 2 standard deviations on magnitude, and we can see that her alpha band is really down. She's down in general, actually, but her alpha band is particularly down. Let's take a look at her. This is a delta phase issues going on in the delta phase, and then we're taking a look here at her beta phase. Real big issues going on with her beta phase. And again, that's really, really where it shows up, is in the beta phase delays in the network. So the signals are not getting across like they should. And so you've got all these perceptual issues going on. This is 18 minutes of SMR beta, and we can see that her alpha waves are starting to really kick in. and where they belong. And yeah, they got really nice and beautiful. And when we look at her now, here's where her alpha is. Before we were, and look at, she's filled in quite nicely across the board overall. This is on microvolts and not on standard deviations. Do I have microvolts up here? This is 1.75 microvolts, so we're zoomed in somewhat on this one. And we can see of course, we can see the OCD down there. I should have shown you that, I guess. For some reason, he hid that slide. Probably because I was talking to another group who didn't really understand EEG just a while ago. So big improvements across the board. Here she is on standard deviations. She's showing a little bit of entrainment at the 20 hertz level, but things have improved quite a bit, and so has the phase. Phase improved a lot. And it's amazing how phase can improve like in half an hour. I'm not sure why it can improve so fast. Maybe it's the lactate and ATP or kicking up the mitochondria. I'm not entirely sure why phase can improve so quickly, but it does. It doesn't hold. It only holds for a couple of days. So that's why people have to take this home and use it every morning for a while. And after, a few months, it will lock in and it will get established. But after this session, that's what she said. My head is clear and I can think. And let's say the effect lasts for about 2 days. We saw her on a Friday, and by Monday, she was struggling again. So we got her device and away we went. Now, this is the SCL9ER here. I didn't process this into those categories like we saw with the other girl, because I'm just showing you what people actually say where they have their issues. So moderate, nervousness and shakiness, unpleasant thoughts, because they're OCD, so stuff is going on in the head all the time. Trouble remembering things. Feeling easily annoyed or irritated is common. These are all very common indicators of this signature. Low in energy, slowed down, blaming yourself, feeling blocked and getting things done, having to do things very slowly to ensure correctness, soreness in your muscles, having to check and double check, difficulty making decisions. That's a big one all the time. Mind going blank, trouble concentrating, heavy feelings in the arms and legs. Well, they're tired all the time. I mean, they're exhausted because you guys are sleeping 3 or 4 hours a night, and it's not good delta sleep either. They're just barely pulling it off. Waking in the mornings. Okay, so what I'm going to do here then, I'll show you a really bad case. I'll show you this guy here, and we could stop with him perhaps. Now, this was the worst case I had seen so far. And this guy, he struggled his whole life. Fogged out, doing poorly in school. He's a loner. He can't connect socially with people. Socially, of course, social connection requires a sharp brain. And he was 31 years old. He'd been into detox. He lost his license for DUIs. He snuck alcohol into detox and rehab. And he's a real binge drinker. He was the first guy to be medevaced out of his little hometown after he was 6 years old and he fell off a slide and he concussed himself pretty severely. What I should at least do is show you the stitches on his head. And you can see, and you can also, unfortunately you can't hear him, but he talks very slowly. He's already showing issues from his concussion. So they gave him a little tour of the fire trucks and stuff, and the paramedics came and rescued him after he fell off the slide. And so he was shut down for 25 years. He is extremely flatlined, and he's a binge alcoholic. So he lives in a basement suite, and a friend is giving him a good deal on a little suite in his house. And he does odd jobs moving furniture for people and, things like this. So he can get enough money to pay his rent, eat a little bit, and buy lots of booze. And anybody you talk to, he's a deadbeat, he's a low life, he's a piece of crap. That's what everybody said about him. Here is his OCD. It's really low. And that's his obsessing on drinking, as his alpha was really knocked out hard. Now, he had such little brain activity that when we did the phase measures, it didn't show up because there were no phase issues because there was no brain activity, basically to speak of. Put him on entrainment, SMR beta. I had him on the alpha beta, for this one. And this is him 15 minutes in and starting to make massive amounts of alpha waves and big delta waves. He slept for half an hour after the session ended. This is his post activity, big improvements. The activity went way up, and he's got fast running alpha wave now, just like we often see. Here he is here. There's his alpha wave there on the database. Now we're starting to see some phase issues, only because his frontal areas were a little slower to respond. So This is him. He quit. So that we saw him in October, like 2017 or 18. He had no, I'll show you that part. This is him. This is him 5 years sober after being a binge alcoholic all his life. And now he's also foreman of a machine shop. And so he slept 30 minutes that night. He slept 11 hours. I mean, after the AV ended, it was 30 minutes. He slept 11 hours that night, continued sleeping well. He had no anxieties. This is 9 days later. No anxieties at all, or no cravings, no anxiety. He was feeling rested and grounded. He felt sharp mentally. He did drink some between October and through Christmas, but never binged. And then he decided for New Year's that he would stop drinking entirely. Now his brain could actually let him do it. Before he was self-medicating. And so these are him. This is a score prior to his pre-score on the SCL90. Same issues as the other lady. This is him after. Look, I used light little check marks now, and he went way, way down. My score is now 10. So 3 years later, I did an EEG on him just to see if it stuck. And look at the beautiful alpha waves he's making. So he used it for about 3 months, and he kind of quit using it. And 3 years later, he is still cranking out beautiful alpha waves. Well, then that's why he can manage to not drink now. So this was his pre, and this is his post right here. This higher voltage. This is where he is now 3 years later. A little bit slow for alpha, but everything is really quite nice. It's where it belongs. And so anyway, here's his phase issues. Really nothing to speak of. So basically, I guess I'll stop there. This was his score 3 years later, 17, and say 50 is the norm. Oh, I haven't even showed you the Alzheimer's stuff. Shoot, I forgot. I'll show you something very fast with Alzheimer's. This was a study done by Leslie Sherlin and Budzinski. This lady had severely declining Alzheimer or progressing Alzheimer's, and she was 57 years old. And they were doing a cognitive decline study, and she got into the group. I guess maybe I opened up too many slides. Anyway, they gave her entrainment. They did Loretta's, and they're looking at, this is delta, and we can see the delta is going down right after their first session. This is after 30. And then she moved out of the senior's home. I mean, here she was shuffling around with severe Alzheimer's. They gave her 6 months to live. She moved back into her own home in Seattle and would walk to the clinic to get upgrades from 6 to 10 month period after her, from 6 to 10 months following, and did really fine. So, and what is so anyway, they did the study. We didn't know what this all meant back in those days, but once I figured it out, I gave Leslie a call. I said, you got to shoot me those files. So he shot me the files, and what we see here when we look at her brainwave, no alpha waves. After entrainment, she had an alpha peak. So we re-established her alpha waves, her glia and dendrocytes. Her glia woke up, and I should show you the glia here. This is the glymphatic system. And when we don't sleep well, the glia does not get, the astrocytes don't work right. They don't pump out all that beta amyloid deposits into the lymph system and get rid of it. So it builds up. So this is what they concluded. She had spatial ability was improved. She was able to find her way around a several block radius, including distance from her home to the site of the training, which she couldn't, before she couldn't find the bathroom in the senior's home. Walking style improved to a normal gait. She reported cognitive fog lifted, and she did really well in the microcog reasoning ability. So this was the first Alzheimer's case that we had actually done. And so there you go. Now, if you want to learn something about Nuka, this is a guy who had paralysis. If you want to do stuff, I talk on a lot of things. This is entrainment. There you go. Want to see what big electrodes do? I'm just kidding. So anyway, there you go. I am done. Thank you very much for attending my lecture. And if anyone wants to do research, especially on some of this stuff for pumping cerebrospinal fluid, that would be awesome. I'm going to stop sharing.
Thank you so much, Dave. We've got a couple questions in the chat. Dr. Andrea had a question about the frequency of entrainment in one of those studies.
It was a study with the students, I believe, and their cognitive function had improved as a result of the entrainment. I was just wondering what the entrainment frequency was.
Oh, typically we're using SMR beta.
Okay.
We put SMR into the left fields of both eyes and the beta in the right fields of both eyes.
Was that the
great.
John Anderson New Horizon study?
I'm not familiar with the New Horizon study. I don't know who that is.
John Anderson?
No, John LeMay. did this study here with P300 responses in college students. That might have been the one you were thinking of.
No, I'm thinking of the one that John Anderson did in the elementary schools in Minnesota, where they used audiovisual entrainment.
That was Michael Joyce who did that study. It wasn't John Anderson. John Anderson did a lot of other things, but he didn't do that study.
I thought he did the EEG side of that.
No, that was I forget the professor's name, but there was a professor there who did the EEG side of that, but it wasn't John. No, but John did.
But that did show changes in the kids who had ADHD.
Oh, huge.
Yeah, huge changes.
Yeah, that's another slideshow. But yeah, it showed huge changes. You bet they improved greatly, and so did college students. Big changes in college students. Victor Wucher did a really good study too. He was out of Germany on memory and concentration, and found that kids get fatigued when they do their post tests, and their concentration and memory go way down. But after entrainment, they maintain the same vigilance they had at the pretest. But yeah, that's a whole other lecture if you guys want to see that one. So, but it is shorter than this one. This is a long one, for sure. So
All right.
Right.
Sorry, we have another question. Kelly Pittman is asking, what AVE program did you use for the Alzheimer's?
Here we go. Sorry, Alzheimer's. Now, Tom's was a little strange. He was randomizing between 8 and 22. And he would hold it for 10, 15 seconds at each frequency, not just randomizing all over the place, which probably wouldn't have worked. We are now using SMR beta. Basically, we use SMR beta for a lot of stuff, for sure. Those bands are certainly, they push up cerebral blood flow. They push up cerebrospinal fluid. They activate lactate and ATP. A lot of things go on. I mean, a lot of these things are kind of generic with entrainment anyway, but certainly the SMR beta band, we use it for ADD, for college students, for Parkinson's, for the seniors with depression, for cognitive decline in general, and also for the Alzheimer's. And we use an alpha beta protocol for the asymmetry type of depression, and that works fairly well. But TDCS works darn well for depression too, but it depends on kind of depression you might have. Sometimes I will do anode cathode, and sometimes I'll do anode shoulder, depending on the type of depression you have and how fear-based it is, or is it just chronic now and not fear-based, and what does the EEG kind of show? So, but that's another lecture altogether. I should show you our new machine that's coming out, and this is it here, and it's called the Premier, and it does entrainment, it does CES, both 50% duty cycle and the short one. It does TDCS. It'll even do low frequency 10s. It's the most advanced brain machine ever made. And we should have it out next month. Colors in the eye sets. You can do any color you want. In fact, we use color to augment the actual frequencies. And you can modify, you can run it off your computer. You can use it for heart rate variability very well. I got a whole lecture on heart rate variability. And John LeMay did some amazing things with people he couldn't train. And when you put them on our breathing app with the lights plugged into a USB port on the computer, instant heart rate variability.
There's another one. Guy Bauman is asking about what we're using for the pumping of the CSF, the cerebral spinal fluid.
What the study did, I'm not doing it yet. I still have to program that into the device. But they tried 4, 8, 12, and 40 hertz. And they found that 8, 12, and 40 had the best effect, but also in 16-second intervals. So 16 seconds on and 16 seconds off. If they were too short, the pumping the cerebral blood flow didn't change enough to really do much of a pump. And if they were too long, they were just plateaued and just sat there. So they could get maximum change at about 16, and they were getting about 8 or 9% exchange in cerebral blood flow every half a minute. Which is a huge amount of exchange in cerebral blood flow. So we could see that as potentially being something very important for Alzheimer's and Parkinson's. We've seen some interesting stuff with our subdelta protocol, which in some ways, which is around half hertz to 1, and in some ways that might be mimicking delta waves, which also pump cerebrospinal fluid. And we need someone to do research.
Fantastic. There's Dr. Andrea, did you want to ask any more questions about using the CES, how frequently you can use the CES on clients?
Yes, I would. I'm curious if it's recommended. It sounds like it is, and I have used it on a couple of clients who are experiencing depressive symptoms after their TBI, and I'm just curious how often I can use it. So I have one client that wants to do it every time she sees me, and like twice a week. So I haven't been doing it that frequently, but
Yes,
yeah,
but CES is a little trickier. First of all, and this is why I should teach you the lecture on NUCA and subluxated atlas bone. Most of the time when people have some kind of a concussion, they have popped their atlas bone out of place and it inflames the brainstem. And the brainstem, 4 neurotransmitters come out of the brainstem: serotonin, acetylcholine, norepinephrine, and dopamine. And I've seen people with concussions who ended up because they inflamed their brainstem and they've overexpressed norepinephrine and their brain waves have sped up to 12 hertz, even though they have their post-concussion. But then you get them into NUCA and you get the neck straightened out, then their brain waves slow down again because the inflammation is down. So CES applied bilaterally across the ears primarily stimulates the brainstem. And so and it primarily increases serotonin and endorphins. Some studies have showed that, in one session, a 20 or 30-minute session, you can get a 200% increase in serotonin and endorphins. So I have found that CES, I've done a few EEGs with CES and pre-post EEGs, and it seems to work best with people who have a fast-running alpha. If it's generalized anxiety and things like this, if it's pain, that's a bit of a different story. If you're using it for pain, then it works not too bad for numbing pain to some degree, but otherwise it's kind of hit and miss. If I'm using on people with generalized anxiety, the depression, first of all, get that neck looked at. That's the first thing you need to look at, especially if they've got postures and they've got all this muscle tension, usually on one side of their shoulder is all in spasm and they got a sore leg because their hip is getting twisted. Get the NUCA done first and then proceed with the CES. Well, you could do the CES at the same time. I guess it wouldn't hurt, but you got to get that brainstem fixed first because it could be over or underexpressing certain neurotransmitters that are leading to the anxiety and the depression. It can be a structural issue, right? So I would do that, and then on the CES side, there's globally CES produces pretty much even in terms of serotonin and endorphins at any frequency. I mean, even the little Fisher-Wallace one is running at 15,000 hertz. Almost all the research is between half hertz and 100, but for whatever reason, they decided to clip it along at 15 kilohertz. I don't know why. And it seems to work reasonably well, too, from the studies that they've done. Otherwise, there's a guy who did a study where he looked at 100 hertz versus half hertz. And he did EEGs, and he concluded that he felt 100 hertz because it increased alpha a little more. He figured that it was probably more effective at increasing serotonin at 100, and delta was improved more on subdelta, which he felt was more of an endorphin response. But it was just one study, and it was loosely based on an EEG. So the general rule of thumb is if it's anxiety and depression and stuff like this, go with 100. And if it's more pain related, go with the delta. But they definitely need more research on that for sure before they would really know. Having said that, there's a study that came out, and it was actually an electroacupuncture study, and they were using the earlobes or right around the earlobes, and they were shooting. They found that half hertz CES was the best frequency for driving up endorphins, which other people have shown. But they also found 100 hertz CES was the best frequency for shooting up dynorphins, which is another set of pain gates. And so, but they found they had to alternate every 3 or 4 seconds between half hertz and 100, half hertz and 100. They also found that 14 had a moderate influence on both pain gates. So in our new machine, we can actually put that, I've actually got that programmed into our new Premier, where we're bouncing between half hertz and 100, half hertz and 100. And then we do 14 for 10 seconds. Then we're doing half hertz, 100, half 100. And it's in a half-minute cycle. So half 100, half 100, like 3 seconds apart, maybe 4 seconds apart. And then a little bit of 14 for about 10 seconds, and then we repeat the cycle. And I'm going to merge that in with alpha protocol with the entrainment and with an alpha beta one as well. So we can also work with general anxiety and work with depression with the light and sound in addition to the cranial electrostim. But haven't wrote that session yet, but I've been meaning to get that into the device before we get it out to the market. So anyway, there you go. There's something there on CES.
Yeah, thank you. Just to add on to that, if a person is taking, like, say, an SSRI, is it unwise to use the CES because it's releasing serotonin, and for serotonin symptom issues, would it be unsafe for them to use it?
No, it would not. It would not be unsafe
Okay.
to use it. People use it all the time on SSRIs. In fact, you'll probably be weaning them off the SSRI to some degree
Okay.
as well. I
Okay.
just found a study. There's a certain herb that people have been taking. What was it now? And it outperformed SSRIs. It was a pretty big study. I remember the name of the herb now, but it was darn. But yeah, you'll be okay with it. Yeah, entrainment as well. Entrainment. We got plenty of people who took entrainment and eventually discontinued their SSRIs.
Thanks so much.
And it doesn't cause any issues. Things like methylene blue, you got to be a little careful with. It can definitely cause a bit of a serotonin syndrome. If you ever decide to stimulate with methylene blue is pretty amazing. It's a nootropic, and it dramatically increases mitochondria activity in the brain. And you got to take it in careful doses, or you could probably overexpress and maybe burn out your mitochondria at the same time if you take too much. When I started first taking methylene blue, I transformed into a 20-year-old like next day. And I just had to run. I had to bike. I had to swim. I had to hop on the motorcycle and do 90 miles an hour down the highway. Oh, it was crazy. But after about 3 days, I started getting exhausted, and I realized I could burn out my mitochondria when I dug into some research. So now I take I cut that dose into a quarter. I spread it over 4 days now, and just gives me great energy. I can just go all day long, and I'm 68, so it makes me feel like a 40-year-old for sure when I take it in proper doses. It's quite good stuff. It is a nootropic. I would say also look for other herbs and supplements that are nootropics, and there's quite a few out there that will actually improve brain function by stimulating mitochondria. Glutathione is another good one. Quercetin, green tea, curcumin. All those things affect mitochondria and help the brain function better. And things as simple as bottled water can be absolutely dangerous to the brain. So you got to avoid stuff too. Very important to avoid all the bad stuff. Don't eat Fruit Loops for breakfast. Eat a decent breakfast. Don't eat fried stuff too much. Don't eat barbecued stuff too much. Really get into raw vegetables and raw fruits. Don't spike your insulin. Insulin can take down eating an ice cream cone can take down your mitochondria for 24 hours.
I think we need to end it here. I know there's still some people asking about autism, and there's some questions.
Okay. Yes, we've done well with that, but that could be maybe another conversation if you feel like bringing me back.
We'd love to have you back for that, for sure. Absolutely. Thank you so much. I think my internet connection is starting to fade, but I really we all appreciate you joining us. And this is the first time we've had 90 minutes, and now I see we need 2 hours for you.
Yeah, I guess I don't normally need 2 hours, but this is a big topic, this one.
Absolutely.
And as you know, I got to do all that physiology to even kind of give you a context of why this is working the way it works. Yeah, and you're right. I guess this is now, I added all that stuff on cerebrospinal fluid and that, and now it's a 2-hour lecture. Good to know.
Yes. Oh, well, thank you. And thank you, everyone. And in November, Lisa Laxko will be speaking also, I believe, on TBI and photobiomodulation. But she's in Australia, so we're going to have to change the time around for next month. And hopefully, Dave, you can join as well.
Sure, you bet. Thank you.
Thank you. Bye-bye, everyone.
Bye-bye.